Stanford University.
We push off into new terrain here, yet another bucket. But before doing that, various bits of feedback from office hours, from hearing from TAs, getting a sense of where the grand gaping craters of confusion are so far. And apparently an awful lot of them were provided in the last 2 days' lectures with behavior genetics. Various issues that came up, clearly one of the most complicated, inaccessible, subtle pain in the neck concepts in the whole class, which is this heritability business. So memorize the following 2 sentences, because it all comes down to the difference between something that is inherited and how heritable a trait is. The fact that humans overwhelmingly have 5 fingers reflects the fact that the number of fingers is an inherited trait. The fact that when there are some circumstances of humans having other than 5 fingers, it is overwhelmingly due to environmental something or others, is an indication of the fact that nonetheless variability around the number of 5 fingers, heritability is essentially 0%. So get those 2 sorted out and you have those 2 concepts all under your belt and very useful. Why is it useful? First off, why is this one useful? Because it is important to know the distinction. Everybody, for one thing, out there tends to view these as telling the same thing. And insofar as they think it says the same thing, they think that both terms are referring to this. Something that is genetically regulated, genetically determined, whatever, 2 totally different terms. So one reason to obsess over this is that when the newspapers give us our soundbite of scientists report that, it's usually this one that they're reporting with one of those marvelously misleading numbers, which then sets people up for thinking they have just been told how much do genes determine some average feature of this trait. So important to tell them apart. The other reason of getting this sorted out is not only to unlearn the nonsense aspects of misinterpreting this term, but because understanding that term gives you a lot of insights into when and how you are getting gene-environment interactions. Caveat with that, saying when and how you're getting gene, you're always getting gene-environment interactions. Remember the quote from the other day, it's like saying which contributes more to the volume of a square or the height or the length or the roundiness or whatever. Yes, it's always gene-environment interactions, saying in what ways are there some of the more interesting ones, dramatic ones, in what realms. Understand this and you avoid this confusion. Understand this and it gives you insights into gene-environment interactions. Okay, other issues that are coming up. By now we've looked at 3 different broad approaches to the biology of social behavior, the evolutionary stuff broadly stated, the molecular, the behavior genetics, and what should be clear by now is if you were living inside one of those buckets, you hate the other 2 and trash them entirely. So we get to the first of our great conflicting points, which is, does that mean one of these is wrong? No, one of them is not wrong, none of them are wrong. Some are more right than others, and some are a lot better than others, but nonetheless, these are all, again, from the very first lecture, different levels of description. Another way to begin to think about this in terms of what we've now been focusing on, and this sort of coming up from some questions afterward. So by now, we've had the term epigenetics. That's coming in the last couple of lectures, and there are at least 3 different levels, 3 different buckets with which you can define the term. What is epigenetics? Epigenetics is the way that culture, environment, all of that affects biology. That's a certain broad way of stating that. Notice stated that way, you were making biology synonymous with genetics, which ain't so, but nonetheless, that's a certain broad level of stating it. In another discipline, a very sort of first pass at molecular stuff, what is epigenetics about? It's the way in which environment turns genes on and off. And at another level of explanation, more reductive, what is epigenetics about? It is regulation of chromatin remodeling and methylation of genes and all of that. Do not panic if that's not the level you want to know it about. That's the business from the other day about changing access of transcription factors to DNA, jargony way of doing it, don't worry. The main point being that this is a completely different level of defining this. This is the whole point in here. We are beginning to see different disciplinary approaches. We are beginning to see where one discipline has decided they've answered a question. This is how culture affects biology. Give me a break. Show me which genes we're talking about. We're talking about genes here. Give me a break. Show me, is this chromatin remodeling? What's the mechanism for it? How reversible is it? One discipline's answers, the next one's starting point, blah, blah. One discipline's wonderfully satisfying scientific answer is the basis for the next discipline to be totally contemptuous of them. You call that science. This is sort of the whole point here, beginning to see how we could chip our way between these. More bits of clarification. Amid that, one of the things that really came through is in the sort of ratio of praising to trashing. I was clearly spending a lot of the last 2 days trashing the behavior genetics approaches. So a little bit of clarification there. First off, we can broadly divide what came the last 2 days into classical behavior genetics, and that's the comparing monozygotic and dizygotic twins. That's the adoption studies. That's the twins adopted at birth. That's all of those approaches there. Those are all the ones where you were just inferring really indirectly that there's something genetically going on there. The other half, and much more sort of the modern behavior genetics, is marrying these traditional approaches with molecular biology. And that was the business at the end. You know the gene, and you kind of have an idea of what it might do. How does that map onto behavioral variability in humans? You know the behavior, and you've got some sense of its variability. How does that map onto variability in genes? This is the much more powerful contemporary end of it. So what is this end good for? Is all it's good for is pronouncing that something's genetic, or it's 73% genetic, and then you trash it because that's gibberish. What it's good for mainly is, okay, so you've got an adoption study, classical behavior genetic study, where they're adopted right at birth within seconds and raised in different households. Oh, you haven't ruled out environment. Don't forget prenatal environment. Oh, you haven't ruled out environment. Remember the non-random assignment of adoption, all of that. Does this mean this approach is useless? No, what it's good for is demonstrating nonetheless, well, we've just ruled out all sorts of realms of environment that people would guess is consequential. It's less consequential than you think. We haven't ruled out environment entirely, and just because it's all gene-environment interactions, we certainly can't come up with a stupid number like that. But what this is good for is at least showing here's domains where people, a lot of people would have assumed there's big environmental effects, much less than you would think. So that's the much more conservative sort of sobrietist thing that people can do with that field. The fact that far too few of them actually do that is reasons to trash it. No, well, nonetheless, there's lots of good things in behavior genetics, but that's the limited domain where it's useful. Okay, so just beginning to get a sense here of the various things that are confusing. Obviously, no one on earth in any exam in here is going to be asked to choose which field is better and which field gives more, you know, kumbaya and all of that, but just recognizing the different approaches and the wonderful rainbow diversity of ways to think about, you know, mating and fruit flies or whatever, and just beginning to see by now, this is what the whole class is about. Speaking of that, I recall from the first class that there's somebody in here who I think was an English lit grad student. If you have a chance, email me about how it's going in here. Let's see, what else? Next week, you know, that I'm curious. I'm very pleased, whoever you are, assuming you haven't fled after the first class, is in here. Let's see, other stuff. Okay, schedule. Next Monday, this coming Monday, we will have a lecture on yet another of our disciplines, ethology, and we will see that's a totally different way of doing it, blah, blah. Wednesday, Friday, and the following Monday are the catch-up lectures in class taught by the TAs. Two of the lectures, Introduction to the Nervous System, the third one, Introduction to Endocrinology. It will be a broad overview. It is explicitly designed for people who have no prior background in this, but what I think is probably a good idea is even if you believe you have prior background in it, maybe assume it might be a good thing to get a refresher on it, and this will be very useful. Following that, we will have two more lectures, more advanced topics and basic features of neuroendocrinology, and then staggering into the midterm, and then, oh, then the second half comes, just a sense of what's coming. Okay, so that's where we're at. So now we transition to the next subject here, which is, no, actually, before I forget, how many, did people see this, the article in the New York Times this morning about the study, the Chutes and Ladders study? Did anybody see it? It was posted. Nobody saw it? This was, you know the game Chutes and Ladders? Okay, we all played Chutes and Ladders. This was this massive study funded by the World Health Organization, where what they did, this is going to be the definitive study on the subject. They showed that people from Nepal are better at playing Chutes and Ladders than are people from Belgium. Cool study. I don't know how you guys missed it. Okay, you need to know about this. This is important. What do you want to know about this study? This massive study shows that people from Nepal are better at Chutes and Ladders than people from Belgium. Ask me questions, since you were terribly, woefully underinformed about this. What more information do you need to be impressed and tell everybody about it in the dorm tonight? What else do you want to know about the study? What does better mean? Okay, social relativist, and does it mean that you learn more about yourself playing Chutes and Ladders? Do it mean that you make the world you win. Okay, so winning is the end point. Good question. What else do you want to know about the study? Yeah. Why? Why, you ask that? Read Aristotle. Since humans first pulled out of the mud, this has been the thing we have wanted to know. And now we do. Son, the difference between knowledge and wisdom. Okay, so why? Okay, what else do you want to know? What other questions? Yeah. The methods, very good. Okay, various methods. Do you want to break it down into more details, given some of the critical tools you have by now in terms of methods before deciding how impressed you are or not with the study? What sort of questions now in the methodological realm? Yeah. Ah, very good question, because we see a first methodological issue. Are you getting a decent sample size so that you're confident that you can actually say something about the population at large? And that's a great question. It was done on everybody in Nepal and everybody in Belgium. Good study. Okay, what else do you want to know? What other questions? Yeah. Well, that's an excellent question. Every single person in the study from both Belgium and Nepal played one game against every single other person in the study. That's why you haven't been hearing much about either country in the news lately. They've been very busy with this study. Okay, so good methodology, large sample size, random assignments of games. What else do you want to know? Yeah.
Why is the question researchers ask worth reading about, and why did they ask?
Okay, that one again. It's self-evident. Know thyself, or know the Nepalese and the Belgians or something. I'm glad I know this now. I'm glad they went and did that. Okay, what else do you want to know? Obviously a matter of subjective taste as to what counts as an important scientific subject. This strikes me as critical. What else do you want to know in terms of what sort of conclusions? They kind of hint, they're not positive, but they're kind of hinting at a genetic component to it. Yeah, question in the back there. Yeah.
What skills are involved in Chutes and Ladders?
What skills are involved? It involves, what is it? They have a whole list there. Let's see. Various tasks involving spatial memory, various tasks involving reversal performance and telekinesis. So it taps into all of those domains. What else do you want to know about the study? Yeah. Is it heritable? Great. Oh, is it heritable? So which are you asking there? Okay, well, they did it. I mean, they are suggesting there's some degree of heritability, and they did it right. They went after all the issues we've learned about by now. Specifically, what I'm saying is that every single person in Nepal and in Belgium was cross-fostered to somebody in Ecuador. In fact, they did it fetally, right after conception. So they can, the prenatal stuff, what else do you want to know in order to decide, am I impressed about the fact that people from Nepal are better at Chutes and Ladders than people from Belgium? Yeah.
Were they all given the exact same amount of Chutes and Ladders as children?
Oh, yes. In fact, none of them had been exposed to it before. They were all raised in hydroponic gardens without Chutes and Ladders, and they were only released in time for doing this. Okay, what else? Yes? Oh, good question. Here we are getting to that, all of that. Everybody got the exact same score from Nepal, and everybody from Belgium got the exact same score. So that's kind of impressive. Are you impressed yet? Are you impressed enough yet to go run out of here screaming with this news? What else do you need to know? Yeah. Who went first? They randomized it. They randomized it by the entire populations played, what is that? Rochambeau, yes. They played Rochambeau, and best of all, they had to play Rochambeau in Esperanto. Yes, more.
What was the environment in which they tested?
Okay, they were released from a hydroponic garden, and then all of them were placed in a completely sterile bubble environment where all they had in there were copies of People magazine in a language they didn't understand. So it was environment was very well controlled for. What else do you need to know?
How much better were they? Like 0.001?
There we have it. They had a huge sample size. They cross-fostered as fetuses. They controlled for environment. They got everybody in there. They did the right techniques. They randomized it, every possible choice. That is so impressive. That is so impressive. How big is the difference? Is it a big difference? And this is a critical thing to start putting there in your armamentarium of critical questions, to start having skeptical ones. Great, you've got a whole bunch of tools by now. Whoa, they said genetic. Did you control for this? Did you control for that? Whoa, they see a difference there. Well, wait a second. Did they all have the same experience? Whoa, did they have a big sample size? All of it covered. Great, wonderful, perfect science. They've got every single bit of it covered there. But then the critical thing you better ask is, how big is the difference? Is it impressive or not? Because we all think we've got the basic tools, or hopefully we all have the basic tools for going at these issues. Okay, was a study done in a way that was clear-cut and objective? Were people blinded to whether or not this person was Nepalese or Belgian, or was there appropriate blinding? Was it done in a way that you can falsify your finding? In a sense, the definition of experimental science. Was it designed in that? Was it independently replicated by anybody else? We all have those tools under our belt by now, but far too often we are not trained at that point to say, wait a second, before I get all excited, how big of a difference was it? Let me give you a real example of this. And this was a paper about 3 years ago in the journal Science, which you should get a sense by now that the journals Science and Nature are the two official biggie ones on this planet in terms of credibility. And this was a paper having to do with IQ. And this was a paper having to do with IQ and birth order. And what they showed in this paper with spectacular statistical confidence and as much of like all the controls in place as you can ask for from the hydroponically gardened fetuses in Nepal, they did absolutely perfectly. What they showed in the definitive study with 250,000 18-year-olds in Norway was that there is a reliable IQ difference depending on whether you are firstborn or latter-born. Okay, how many of you are firstborn? Whoa. How many of you are only childs? Okay. How many of you are number two? How many of you are more than number two? Okay, so how many of you think the highest IQ comes from the firstborn? Okay, and we'll assume the converse with the other group. Did anybody just vote against their own birth order? Whoa, okay. Well, that's impressive. We salute you. Okay, so they found a difference. They found a difference which is firstborns, in this very statistically reliable way, have higher IQs than latterborns. They restricted the analysis just to secondborns to keep things clearer, and they showed firstborns have higher IQ than secondborns. 250,000 people, as close as you can get to all the people from Nepal and Belgium. Huge sample size. So they report this was the most thorough study ever done. So what sort of questions do you want to ask? Give me hypotheses for what's going on. Obviously, sort of one is that like the parents pour protein into the ears of firstborn and the second one just gets Fritos or something. But give me other hypotheses for what could be happening. Possible explanation for this in terms of biological, socio-cultural, endo-immuno, psycho, yeah.
I think we already know that firstborns tend to be more like, well, I mean, it's like the parents, they're more worried about making mistakes and they kind of raise them harder, so to speak, and then they sort of relax in the second-born child. So we already know that firstborns tend to go to college and stuff like that, do more traditional, successful.
Okay, so great hypothesis. It's more parental investment in the firstborn, and those are the ones where the parents freak out with everything, and by the time there's the third one, they're all foraging on their own when they're 6 months old. Okay, so parental investment. They went after that one correctly. Here's what they showed to rule that out, which was if you are an only child, you have a lower IQ on the average, blah, blah, than firstborns who have younger siblings. So it's not parental investment, it's something about being the firstborn of multiples. Yeah? Okay, so pressure on the firstborns to be firstborn, how would that raise IQ? Okay, so that's one of the models out there. A variant on that is firstborns get pushed into a tutoring position early on the well-known fact that occasionally now and then teaching something actually causes you to know what you're talking about. So the firstborns, because of this tutoring role, oh, that would control for the single child versus the firstborn difference there. So maybe it's a tutoring phenomenon. What else could be happening? What other possible notions? Yeah?
It depends what age you're looking at the kids at, because if you're looking at like actual children and comparing IQs, older children will probably have higher IQs because they've had longer time in life to learn.
Okay, great idea. Up to age 12, latterborns tend to have higher IQs than firstborns. By age 18, which is when this was studied, was carried out, it flips the other way around. So any hypotheses for why this difference occurs? Why by 12 years of age, latterborns tend to have higher IQs than firstborns? Why does it then flip afterward? Yeah. Okay, so is it IQ testing, some biases with that? What would rule against that, though, is that the age controlling for only children versus firstborns. So that would definitely be a possibility. That was ruled out. Yeah.
I think that I've learned in other classes they said it has to do with the ratio of adults in the environment. So when there's only one child, you have more adults with higher language skills.
Good. Okay, so another version of a parental investment model there, which is the fewer the children, the more parental energy. So again, that's one of the standard things in the field. They ruled that out with comparing the only children versus firstborn child. So that's been a dominant model in the field, so they had good data against that. Yeah?
The age of the mother when the baby is in fetus.
Oh, okay. What's your idea about that?
Well, younger mothers, maybe the fetal environment is better, and obviously older children have younger mothers than their siblings who the mother was older when she had them.
Okay, so we've got an intrauterine effect. So we've got egg quality and age of mom and all of that. They controlled for that, the age of the mother. Can anybody think of something, another intrauterine mechanism, though, for what's the difference between being the first fetus who hits the womb of your mother versus being the second or third or fourth? What's one of the biological things that might happen? Yeah.
Stress levels.
Okay, stress levels, which is a way of stating something about intrauterine environment. The more times you've done this, perhaps the more stressed you are in there. What else could biologically, that's definitely one of the things, what else biologically can happen over time? Remember progesterone the other day from that lecture of making new if-then clauses and glucocorticoids, and this is a bit of a stretch. This takes some sort of fair amount of OB/GYN knowledge. What's a danger as you have more and more pregnancies in terms of your immune system? Yeah, wait, I heard that mumbled. Sure. Yeah, okay. Immune suppression, so mom could be getting a lot less healthy, which is another version of tapping into that notion there in terms of egg quality with age, separative age, the number of times you've gone through this. What could be another possibility immunologically with repeated pregnancies? Yeah? Okay, despite that immune suppression, on the average, with later pregnancies, you have a greater likelihood of having formed antibodies against aspects of the fetus. They controlled for that. They showed if you were second born, and there were kids after that, if you were second born and the firstborn died, you reverted to the firstborn IQ, showing that it was not anything about, oh, you were the second fetus in there with more antibodies. They controlled for that. That's actually one of the ideas they brought up in there. What else? How about that business about up to age 12, the second born does better. After that, by age 18, the firstborn does better. Any theories with that? Yeah.
Could it have to do maybe with like how fast they grow or something? I mean, we talked about how like for fathers, it's like better if the child grows faster. Maybe it's like if you're born later, you grow faster, but then like that takes a toll on your like sexual health.
Okay, so an early advantage and you pay for it later type deal. So that's a possibility. What else could come in? Think about, back to the idea there, about if you're a firstborn, some of the firstborn responsibility stuff, how that plays out in family dynamics early on. Why are the secondborns doing better in the first dozen years? Why does it reverse later on? More ideas about that. Yeah.
The second-born benefits from the tutelage of the firstborn early in life, but later on having the older child having had that experience of being responsible benefits from that.
Exactly. That's one of the main theories. That's probably the predominant one proposed to explain that age switch. You get the second kid show up, and suddenly you have a neotenized, dumbed down environment where suddenly the 8-year-old kid is watching Tinky Winky again for the first time in 6 years. That it's an environment that's then dominated more by having a younger one around, and what the older one is mostly doing is the tutoring, and it takes a number of years for the advantages of that to finally come through. That's the main model that's given for that. Any other ideas? How about parental resource stuff? The fact that the larger the family, on the average, in westernized countries, the lower the socioeconomic status. Run with that one. Where does that fit in? What else? What else could be happening with that? Yeah.
If the socioeconomic status of the family is lower, then that suggests that the children might bear more burden in some respects, with more children around, so they have to grow up more quickly and get exposed to the real world. Which is
Good. It's the later ones who are out hunting squirrels and not getting the violin lessons. That's another version of the parental resource one. One being because there's a smaller ratio of parents to kids. The other being because the more kids, the more expensive for the same parental income. You have got less to spread to each child, so they controlled for that, looking at within family rather than just between family. They covered all of this. This is going to be the definitive study for the rest of all of time, showing what's going on with IQ by birth order in 18-year-olds in Norway in 2007. And essentially, what was the magnitude of the difference for this study? 2.3 IQ points. And thus, coming back to what could have been the very first question sitting there when these guys were ready to announce this to the world and sort of start, selling their Be Like a Firstborn IQ self-help books and all of that, lost in there, and this was picked up all over the press and no doubt endless snotty comments by David Letterman or what, and nobody, 2.3 IQ points. You sneeze while you're taking an IQ test and have to wipe your nose for 8 seconds afterward, and that's going to cost you 2.3 IQ points because you get distracted for a second. An amazing example of this whole business of, yes, impeccable science that these people did phenomenal. I don't know who possibly gave them the money to do a study like this, and at the end of the day, totally cool, irrefutable, statistically totally reliable, which is very different from saying important. But what you got at the end of the day was this mammoth study producing 2.3 difference. And this is a great demonstration. The difference between how solid the science is, how confident you are of the finding, which takes in all the variables of sample size and objectivity, how confident you are of the finding, and how big of a finding it is, and those could be two entire differences. So as a prelude to all the stuff that's going to come in the second half of the course, one of the next tools you have to have in mind, in addition to all the ones that were apparent here in the questions you were asking, another one is to keep saying, well, is this a big effect? Is this a reliable effect? Is different from, is this a big effect? Is this a consequential one? So another tool to have in hand. So with that in hand, go and tell everybody tonight about chutes and ladders. Okay, so what we jump to now is something that's been running through a whole bunch of the lectures already. Okay, we've got all those evolutionary models of individual selection and kin selection and in-group and out-group, and we've got something about the molecular biology of why it is that you share 50% of your genes with this relative and 25 and 12 and a half and all of that. And somewhere in there, lurking through all of it, is a question, which is what we'll focus on now, which is how do organisms, how do animals, how do individuals recognize relatives? Because you can't do any of that evolutionary biology, kin selection theory stuff where it's all predicated on degree of relatedness unless you know how related am I with this individual. So what we're going to be looking at here is why or how. Sure, let's go for how instead of why. How do animals, how do social animals recognize kin? What's clear is it does not take a very fancy organism. And there was a great example of this, which I think I stuck in the extended notes last minute, a paper published just a couple of weeks ago, looking at deer mice. Deer mice, and much like their vole cousins, if they are cousins, some deer mice strains are monogamous and some are polygamous. This appears to be a frequent theme in these little rodent things. And with the deer mice, what you find is with the polygamous ones, one female mates with a bunch of males, and as a result, one female will have sperm from a number of different males afterward in the vagina, and what you get is evolutionarily, from all the rules we learned by now, perfectly logically, you get intrasexual competition between the sperm from the different males. We already heard a version of that with the flies back the other week there, where the sperm of one releases toxins that kill the other sperm, but in the process could damage the female's future. All of that, this is a theme that runs through a lot of the literature on sexual competition. Sperm competition, and there's even hints that something like that goes on in humans. Okay, so what form does it take in these deer mice? As follows. I don't begin to understand the mechanics of this, nor do I want to, but apparently with deer mice sperm, if they all clump together, you know, many hands on the oars or something, if they all clump together, you get this macro sperm thing, which swims upstream faster. And the paper had all sorts of diagrams of this, which I did not want to look at in much detail. But there's this, so you've got with the polygamous strains, you've got this problem. If your sperm want to do things absolutely correctly, they only want to form one of these big old, you know, pleasure boat aggregate crew things with sperm from themselves. With sperm from only themselves, and following all of our theorizing, to a lesser extent with close relatives, and not at all with sperm from some other guy. And that's precisely what they showed in this paper. You take sperm from monogamous strains and you put sperm from different males together and they all happily form this big cooperative clump of sperm there. But you take them from species that have evolved under the selective pressure of polygamy, and the sperm there know who they're related to and will form these clumps only with the ones from themselves. You can immediately design all sorts of lock and key stories for how that's pulled off. You could immediately come up with some approximation of what the molecular mechanism would be. But for first pass, what's striking here is, ooh, how do organisms recognize relatives? There are out there single cells that can do this under exactly the evolutionary circumstances models that we've got already. So as we begin to look now at how whole organisms do it, even cells can do it. And we're going to see lots of different possible mechanisms. In lots of species, what you have is some equivalent of what those single sperm are doing, which is there is innate recognition of relatives. How do you show this? We already know the classic ways of doing this, which is the crossed fostering approach. You take a litter, newborn litter, and your rodents, and you cross-foster them to other females. And if later on they can behaviorally differentiate between their siblings and non-siblings, there's something innate about it. Oh, wait a second, what about prenatal environment? Wasn't there something about... So now you do the prenatal cross-fostering, the fetal transplant approach, and you get the exact same thing. There is innate recognition of relatedness in all sorts of rodent species. Another way of doing it even cleaner. You have 2 different litters from the same mother and father rodent, and then they meet together, so there was no shared prenatal environment, and you can show recognition there. You put the rat later on into a cage where there's the urine of its sibling and there's a urine of the perfect stranger, and they will prefer going to that one. You could show that it's even more subtle than that. They will prefer to go to the urine of a full sibling versus a half sibling versus a first cousin, all the way on down. They could take it out to about 3rd or 4th cousins. Incredible discrimination there that can go into it, and it has to be that way, or else all of the theorizing from the other day, you can't figure out how to give up your life for 2 cousins or 8 brothers or 2 cousins or whatever the math is, unless you know who's who. And in some species where it's done entirely instinctually, that would be the way you demonstrate it. So what's the mechanism there in those cases? The most studied ones are olfactory signatures. Pheromones, we've already heard that term in here, and we're going to hear tons more about it, but pheromonal communication. What does that begin to require? If you have pheromones, odorants coming out of, say, the urine from different individuals telling you your degree of relatedness to them, it requires 2 things. It requires qualitative differences in the urine, reflecting the genetic makeup of the individual who provided that urine to the grad student, and it requires some mechanism, some olfactory brain processing mechanism to be able to pick up whatever those differences are. And both have been shown. Okay, the way you begin to get the differentiation at the end of how the urine smells differently. What you've got, and referred to back with the transposable stuff, is you remember in vertebrates, you've got some of your highest rates of transposable events in your immune system, your genes devoted to immunity, where you juggle them around, and that's how, with any luck, you come up with an antibody that will recognize this completely novel pathogen. All of that, there's an additional stretch of genes in that neighborhood where what happens with that is it also undergoes huge amounts of splicing and transposition and juggling and all of that, and what you do then is you create a completely unique protein. You do it in enough of a combinatorial way that it would take, statistically 400 quadrillion Google Flex whatevers to come up with another organism with the exact same protein signature. When you make a protein from that, you have made up one that no other organism on Earth has with a very high statistical reliability. This is a stretch of genes called the major histocompatibility complex, MHCs. And what you see with those is histocompatibility, that whole business with organ transplants, how compatible of a donor is it, how closely related, how much of this jargon, for those of you who know it, how much of a shared antigenic determinant, how genetically similar is this fingerprint, this identifying ID of a protein that determines things like histocompatibility, how well organ transfers work. That's the origins of the term. So every single one of us, every single organism out there has made a arguably unique juggle of these genes and comes up with this signature protein that it sticks on the surface of every single one of its cells. What's that good for? That's good for your immune system learning, if we run into a cell with one of those things, it's us. Don't attack it. Don't form antibodies against it. And if we run into anything else in here that doesn't have one of those, it's an invasive pathogen, and go attack it. This is the basis of the self-non-self recognition ability of the immune system. And what autoimmune diseases are is when your immune system screws up and begins to mistake one of your signature proteins, your major histocompatibility gene-derived signature protein, as in fact being invasive. And one of the other things you hear about, the other day we heard about this tropical parasite, trypanosome. What it does, as you heard, was it keeps juggling its surface proteins. So just as your immune system is all set to attack it because it's got antibodies to recognize it, it has changed its signature protein. There's another tropical parasite, schistosomes, where what they do is they steal your major histocompatibility proteins from the surface of some of your cells and glue it on themselves, and they are wolves and sheep major histocompatibility proteins or some such things. So that's one major domain where this unique protein derived from a unique gene, unique protein signature lets your immune system work properly. So it turns out there's another whole domain with it, which is these proteins can become soluble, which means they're no longer anchored to a cell, they're just floating around. And ultimately, they're floating around in your saliva, in your urine, in your armpit exudates, or whatever it is. And what they begin to do, complicated mechanisms, which I think I will bypass, what they do is give a unique signature to the pheromones coming off of you. And as we will see in lectures to come, animals of all sorts of species can tell, is this individual of my species? Are they the same gender? Or are they an adult? Are they sexually mature? Or are they healthy? Or are they pregnant? Whatever. But thanks to this major histocompatibility business, they can also tell, is this a relative? Now, the juggling events, the splicing and the juggling of the genes, has some degree of statistical relatedness the more closely related you are. In other words, you smell your own urine, if that's your hobby, and the major histocompatibility genes in there will obviously exactly match your own. You smell those of a full sibling, even more questions to be asked, and you do that, and there will be a greater degree, statistically, of similarity of the structure of that protein than with a second cousin, than with a perfect stranger, than with a Nepalese if you're from Belgium, whatever it is. What you find in those cases is that's how you not only get innate olfactory recognition of is this a relative or not, but how related of a relative. So that's half of it. That's how you generate the unique signature at the olfaction, at the pheromone end. The other trick is, how do you generate an olfactory system that could make that discrimination? And all we got to go with at that point is, you can guess, if you think about it a bit, is we got to have some version of olfactory receptors that do the old lock and key business. Just as you make a protein, which has a certain shape, indicating that this is your unique signature. What you want to do is have receptors that will have the uniquely complementary shape for the lock and key, so that you can do oh, this fits perfectly. Let's transduce this to a signal to the brain saying, I'm smelling my armpit. And if instead you've got a protein that fits in there like a lock and key, but not quite as well, you send the message of, oh, I'm smelling my sibling's armpit. And if it fits in there not quite as well, and all the way down, you could begin to see exactly how you designed this. If you've got 1,000 of these receptors of this shape, and every single one of them it hasn't fitting in well enough to stay in there for 3 seconds, so that all 1,000 of them send the signal, it takes 3 seconds of binding there to cause the signal to happen, all 1,000 of them send the signal, it means it's me. It doesn't fit quite as well, so statistically only 800 of them stay in there for 3 seconds, so 800 of the cells are reporting, oh, that's a full sibling. I don't know if that's the mechanism, but this would be a way of constructing it. That's exactly how it could be, along the lines of lock and key, genes produce proteins of certain shapes, certain functions, all of that, and that's how you begin to do it. What have we just gotten? We've gotten a protein, a molecular basis of our theorizing the other day of an if-then clause. If and only if this is a close relative, then send the message to the neurons that do cooperation. We already know that's gibberish to say that there's neurons that do cooperation, but you could begin to see how this is going to work. This is the if part of all the conditional if-then clauses built around degree of relatedness. So how does your olfactory bulb do this? Very interestingly, people are beginning to get a sense of 2 hormones that are relevant to this. One is called oxytocin, and the other is called vasopressin. And what we will see in lectures to come is, particularly in females, oxytocin has long been known to play some like plumbing nuts and bolts job in giving birth, and vasopressin and uterine contractions, and take your average like off-the-rack endocrinologist, and what these hormones are about is like your uterus contracting and giving birth. But oh, that's so little of what they do. What they're much more interestingly involved with is what happens next, which is now beginning to learn how to recognize the smell of the individual you just gave birth to. Because it turns out what oxytocin and vasopressin do in the olfactory bulbs, the olfactory system, the olfactory equivalent of your eyes and ears, is they tune up the cells that recognize major histocompatibility signals. They make them attuned to, is this a relative or not? And there's an if-then clause. If the levels of this hormone have the certain high levels indicating that I just gave birth, and I smell something whose signature odorants fit really well into this receptor, then this is someone who I'm going to nurse like crazy unless it turns out to be my mother or grandmother. Okay, let's put another. If they're very little and cutesy and make cute little mewling sounds, then I will take care of them and nurse them and all that sort of thing. And it's turning out that that's what oxytocin and vasopressin are doing there. You generate mice with genetically knocking out oxytocin or vasopressin, or their receptors. If these are totally new terms, this will come by the week after next as an introduction. You knock out those genes and you get what is called a social anosmia. Anosmia is the inability to smell something. A social anosmia is your nose is working just fine. If you're a rat, you can discriminate different food types, completely arbitrary odors. You just can't distinguish between individuals. And there was a paper a couple of weeks ago showing for the first time what the model has always been is that circulating oxytocin and vasopressin get into your olfactory system and have those effects. What this paper showed for the first time is you're making those hormones right in your nostrils to begin with. This is what tunes it up. Very interestingly, there's also a literature emerging suggesting mutations in genes relevant to oxytocin and vasopressin in families with a high incidence of autism. Autism, a disease where one way of characterizing it is there are enormous deficits in normal socialization, interaction, social bonding, social affiliation, and this suggests that has something to do with it. Okay, so that's a first mechanism. That's how it might work innately. Very cool study in recent years, also showing one facet of this. One of the things you get taught in intro neuro, if you've taken it any time in the last 5,000 years, is when you've got an adult brain, it doesn't make any more neurons. You've got all the neurons you're ever going to get by the time you're 3 years old, and all you can do thereafter is squander them away on like stupid weekend binges or whatever. And it turns out that nonetheless, this is not true. And arguably, this is the biggest revolution in neuroscience in the last decade or so, adult neurogenesis. And it turns out this adult neurogenesis happens in only 2 areas in the brain. The first one is really interesting because it's this part of the brain called the hippocampus. Hippocampus, learning, memory, it's totally cool. And a gazillion studies now showing stuff like you learn a new fact. You stimulate neurogenesis in your hippocampus. You get put in an enriched environment. You exercise. You do all sorts of stuff. You make new neurons there. You get stressed, you make less new neurons there. This whole new field, and 99% of the studies have been about these populations of neural stem cells in the hippocampus. Totally ignored has been the second little pocket of these neural stem cells that can make new neurons. Nobody's interested in it. What's this about? It's totally boring. Where's the second pocket? Just behind the olfactory bulb. And what this study showed, and this is one in your reader, just at the abstract, what it showed was if you have a rat, right around the time she gets pregnant, she starts making new neurons out the wazoo, not from the exciting hippocampal pocket, but from this boring little olfactory end. What goes on with the onset of pregnancy? Female rodents do this massive renovation job of all their olfactory neurons going on there. What it is, they showed in the study, it is driven by the prolactin levels that rise during pregnancy. And what have you got there? What they showed was right around the time she gives birth, she's got this spanking new, completely renovated olfactory system. just in time to do one of the most important olfactory things of her life, which is quickly figure out which ones are her babies, to quickly do the social bonding to them. What hasn't been tested yet, but what I guarantee has to be there, is that vasopressin and oxytocin has to have some sort of role going on in there. Really interesting. Interesting implication of this. So think about this. If you were pregnant, and assuming this works in mammals other than rodents, and if you were pregnant, so the whole time you were pregnant, what's going on? You're doing this huge job of ripping out, you know, the walls and the plumbing in your olfactory bulb, and like putting in new stuff, and it's a total mess. Like you spend your pregnancy with your olfactory system totally cockeyed. No wonder stuff smells weird, and no wonder foods taste weird and all of that. There is a whole adaptationist literature out there on why should it be that you suddenly want pickles and these foods you can't stand the taste of, and it's to avoid inadvertently eating toxins. A really unconvincing spandrel-filled literature out there. It may be an inadvertent spandrel byproduct of you're ripping apart the whole olfactory system so you're all set to recognize the smell of your kids. You just got screwy olfaction and taste all throughout pregnancy. The main point of this, though, is this is endocrine regulation driving, not to make you able to recognize a relative, because you've already got the genes in place for that, just making sure that your olfactory bulb is at the very best at that time for doing it. Okay. What else does one want to know about that? Okay, one additional thing you could do with that information, which is, okay, so why do you want to know who your relatives are? It's who you mate with, it's who you cooperate with, it's who you try to kill, it's who you take care of, it's who all that sort of thing, all of these domains. Also, it's who you pay attention to socially in terms of gossip and such. One interesting study that was shown, which was with baboons, and this was those same folks at University of Pennsylvania. What they did was they recorded the voices. You notice this business about playing the voices of some animal in the bushes and looking at the response of everybody else is one of these standard tools. What they did in this case was the voice of two animals from that group, from that troop, and what was heard was the voice of the lower ranking animal giving a dominating vocalization, and the voice of the lower ranking individual giving this terrified subordinating noise. So they obviously were not getting a terrified subordinating noise out of number one, but they had to sit around and get recordings of number two and everybody else at some point or other. So now they could put them down there, and they would play this, so everybody else is sitting there and saying, what? Number 4 is terrified of number 27. What's going on? And what they showed was everybody paid a huge attention to this. If they were hearing number 27 was trashing number 4 if they weren't relatives, but if they were in the same family and there was this dominance reversal, nobody paid attention. Crazy relatives, who knows what's going on in that family? They're just squabbling. They distinguish the social implications of a dominance reversal depending on relatedness or not. One additional thing with it, before we go to our next way of recognizing relatives after the break, hold on. One additional thing is, of course, telling you who to mate with. And it's obvious who you're supposed to mate with in species after species, someone who is not related to you, because if you do, you may inadvertently produce babies with 2 tails and 7 fingers and all of that. Nobody picked up on the fact that actually we have 10 fingers instead of 5, but we'll let that slide. But what you get there is, oh, avoid inbreeding, don't breed with relatives. Well, we've already heard about a counter-argument, which is breed with relatives because of the inclusive fitness, the kin selection advantages of doing that. And from the first minute people started getting these theoretical models, it was clear that in fact there were contradictory pulls between do major outbreeding in your mating and doing inbreeding with your mating. And people did all these theoretical models of econometrics of where you optimize the different, and they came out with the conclusion that in all sorts of species, the optimal balance of avoiding inbreeding disasters with advantages of kin selection would be to mate with something like a third cousin. And you go and look at all sorts of species out there, and that's precisely what they do. That's where it balances out, and again, you can't do that unless you know relatedness. Then, a couple of years ago, along came this researcher, Martha McClintock, University of Chicago. She's the person who discovered the Wellesley effect back when. And for all of you guys who are gearing up for senior honors theses, this was her senior honors thesis, discovering the Wellesley effect. So this was like one impressive study. Yeah?
Did Darwin marry a second cousin?
Who did he married a first cousin? First cousin. There you go. When we all know he would much rather have married a Galapagos tortoise, but his parents forced him. Well, interesting. Okay, so you thus catapult us here into the human realm. What McClintock did was a study a couple of years ago where she took swabs. She has this whole paradigm of very high-tech, getting cotton balls and rubbing it on people's armpits and putting it in a jar there, and then getting these volunteers who can smell it and give some assessment of how good it smells or not, which in and of itself is pretty wild. And you run that with humans, and of a curve of relatedness, whose odor gets rated as having the most appealing? Third cousins. Yuck. Think about that one later. We are just another species in that regard. All of this suggesting, even in humans, you are balancing this disadvantages of inbreeding with the kin selection advantages. And again, there's no way you can do that without knowing who's related to what extent. Okay, 5-minute break, and what we'll then transition to are species that don't do it innately, but instead have to imprint early on after encountering them. Okay, let's get going again. Let's see, first off, thanks to the wonders of Wikipedia never being that far away from us, we now know here were Charles Darwin's parents, who were third cousins, and then Charles Darwin married his first cousin. So there you have it, something or other, but this apparently was rather common at the time and is not quite the optimal according to Martha McClintock. Okay, so pushing on. So we've now seen why you would want to recognize your relatives and recognize the degree of relatedness, all of our models of who to compete with, who to mate with, who to nurse, who to take care of, who to be voyeuristic about. And we've seen the first domain where you can do that, which is to recognize somebody innately. And one very confusing aspect of it, which I managed to make confusing, is so what's up with olfaction with that and oxytocin and vasopressin and prolactin? Okay, as follows. It is innate that you will have receptors, olfactory receptors in your olfactory neurons, in your olfactory bulb, in your nose. It is innate that you will have olfactory receptors that will be able to detect degree of relatedness, how close an olfactory signature is to your own. That is innate. That will be there. What appears to be the case is oxytocin and vasopressin make you more likely to make those receptors, increase the number of those receptors. So this is not oxytocin and vasopressin making you suddenly be able to recognize relatives. It's just making you better at doing that, more sensitized to it, so that you have 100 receptors reporting instead of 10 of them, more accuracy. What prolactin appears to be doing is, and this is understudied, but the best bet is this is another way of getting more of these olfactory receptors online right around birth to innately recognize your relative. In this time, instead of making neurons make more copies of the receptors, it's making more neurons. It's going to be more subtlety than that, but broadly, those are two different ways of making you better at doing something that is innate in you, rather than making you suddenly able to learn how to do this. So we now transition to the second way in which relatives are recognized, where it's not innate. It requires imprinting. It requires some sort of learning which leaves a long-lasting message of it. Imprinting. So now we got imprinting in yet another use of the word, and major use of it come next Monday. Imprinting, how an animal learns who its mother is, how a mother learns who its babies are, how it imprints on the smell, on the sound, on the whatever of its offspring or parent, how that learning goes on. What is clear is that's a case where the learning occurs at that point is innate. What is learned is experiential. Important sort of distinction there. Okay, so what goes on with this? So in lots of species, you learn the sound of your infant's voice. In lots of species, you learn the odor. In lots of species, you learn what they look like. Different species, different modalities that dominate. What goes into, for example, learning what your offspring smells like? Remember, it's not innate in these cases. For example, a goat, a sheep, whatever, they do not innately have the means of recognizing, oh, this is somebody whose major histocompatibility proteins are fairly similar to mine. In fact, they share 50% homology. Oh, this must be my child. That's not done that way. These are not innate cases. So what sort of rules might you have for how to recognize your offspring? Here's a simple one. Okay, there's a whole bunch of babies out here, and which ones are mine? And I'm a goat trying to figure out which one I'm going to nurse. What would be a good rule that is not innate? Instead, building on learning something at that point, I know I'm going to start taking care of whichever kid there smells like my vaginal fluids. That's a pretty reliable way of figuring out this is somebody you just gave birth to. Or this is someone who I lick as they're first coming out, and for a while afterward, I figure out who am I nice to? Someone who smells like my saliva. Someone who I scent mark right after this is someone who smells like my whatever glands I'm using there. This is someone who smells like my amniotic fluid. This is someone who, after I get that learned, I then nurse them for the first time. This is someone whose mouth smells like my milk. You could see now in this case, it is learning being built on top of whatever your own recognition system is of smell. You can have elaborations on this. How do you recognize a sibling in species where it is learned in this imprinting way? Oh, I'm going to imprint as a relative on somebody who smells just like mom. Somebody who smells like mom's vaginal fluid or her saliva or any of that same stuff going on there. Or I'm going to be nice to someone who smells like someone I mated with back when. That's another strategy. In various species, you could begin to see how all of these are ways of just getting logical information. Someone who has the voice like someone who I heard peeping when they were still inside the egg. Oh, it's them. I'm going to nurse. No, I'm not going to nurse them. I'm going to give them worms or whatever it is birds do. But all these ways of using sensory information to say, oh, that's the one, that's how I know it's them. So all sorts of ways in which this could be done. How can you prove that this is not hardwired, this is not absolutely dominating? A technique we've been hearing about already, which is that cross-fostering business, which is you take newborn whatevers and you switch mothers on them, and the mothers will take care of them. What does that tell you? It means other attributes of these pups coming over to them, these rat pups, override the fact, wait, these kids don't smell like my vaginal fluid. Wow, they're cute though, and there's no other moms around, and they sure look cuddly, and so maybe it takes you 10 seconds to decide this is one of mine instead of 3 seconds. You have different things being played out there, contrasting sort of signals coming through. So that's another domain of doing that. So now we move to us and how we do recognizing relatives. And initially, what the answer seems to be is that we have a different version of it. We do not recognize relatives innately, nor do we recognize relatives by imprinting. We don't do that deal of like we smell our parents right after they're born, or we sniff the vaginal fluid and that's how we know who mom is forever after, or stuff like that. So we don't, instead what do? We do it cognitively. We figure it out. We think about it. We think about it with active, conscious, cognitive rules of how you know a relative is. And of course, what we're going to see shortly is that's not really how it works a lot of the time. But for a first pass, we do it cognitively. So now you do it instead of, this is someone who smells just like my vaginal fluid, the way the goats are doing, or this is someone who I innately recognize as my child, the way the rats are doing it. You're saying, well, that's the baby I just gave birth to, because they haven't taken him out of the room yet, so that must be a cognitive strategy. How do I know who the father is? Not because I can smell in the babies 50% sharing their major histocompatibility gene. This is the only person I had sex with during the cycle that I conceived. a cognitive strategy. This is what humans do again and again because we can think. We can go through stuff like that. We can also do other versions of it, which is, well, this is someone who I saw mom give birth to. Or at least when they took me out of the room and then they like gave me some stuffed animals to keep me from like getting too upset and they said, here's your new baby, whatever. Oh, okay, which is a variant on this is someone who's been around ever since I saw mom give birth to that individual. This is someone who looks like me. This is someone who looks like a family member. All of these cognitive strategies going into that. We think about it. We think about it. And it turns out we have brain mechanisms that are good for doing that too. But we're not the only species that thinks about it in that way. For example, baboons, wonderful study a few years ago, baboons can do this with some sort of statistical thinking. Okay, baboons are polygamous. Females will mate with a bunch of different males during her cycle and conceive, and thus it's not at all clear who the father is, but everybody there plays a guessing game and everybody does some statistics. The usual rule is baboons, being a highly tournament species, males do no parental care of offspring. Turns out that's not quite the case. Some males do, when they're pretty sure who their kid is. And here's how it's done. You are a baboon and you are just hitting puberty, you're female, and what goes on is for like your first half dozen cycles, you're cycling with, you're probably not ovulating yet, you're not quite fertile, this happens in humans as well, and none of the big high-ranking guys are terribly interested in you. Probably because you're not pumping out a whole lot of interesting pheromones yet. So who do you wind up with? You wind up with some poor like adolescent schnook who has no chance to mate with anybody else, and nobody's contesting his ability to have a consortship with you. So you do all of your mating with him. The vast majority of the time they're not fertile because you're not really off. Every now and then though, you get this like junior high school baboon guy who gets his girlfriend pregnant and he's the only one, and what tends to happen then is when she gives birth, he gives a fair amount of parental care to the offspring. So what does he have as a rule there? If I'm the only one who mated with her, if they're using a cognitive strategy, then I'll take care of the kid to some extent. And it is a sight to behold at how incredibly inept an adolescent male baboon is when he's trying to be paternal. But what you wind up seeing there is, well, maybe he's imprinted, maybe he's doing major histocompatibility, gene innate stuff, or maybe he's actually thinking, hey, I was with her 24/7, so it's gotta be me, I'll take care of the kid. Now you see the more complicated circumstance where it's a more desirable female, more mature one, where there will be a bunch of males contesting. And what you will tend to see is 2 days before and after she ovulates, she'll be mating with number 10. A day before and after, she's mating with number 3. The day of ovulation, she's mating with number 1. That tends to be the pattern. Stay tuned, it doesn't fit that perfectly because female baboons also have some opinions about who they want to mate with. But nonetheless, there winds up being this pattern, just like that. So there winds up being this pattern, and thus you're the male afterward when she gives birth and you're trying to decide, is this my kid? Or should I give some male parental investment? And what is shown is baboon males do statistics. They do probability. What they do is, if this was a male who was mating during her prime ovulatory day, he is more likely to take care of the kid than a male who was mating during this window or this window. They are playing probability. They're not very good at it, no surprise, but nonetheless this pattern emerges. That's not innate recognition. That's not innate recognition of some smell. That's just thinking through it. Was I with her? Yeah, but she sure smelled a lot better the day after, and that other guy was with her, so I guess I'm in this category. Okay, well, I'll smile at the kid now and then and tell them I like their piano playing, but I'm not gonna like invest anything kind of stuff there. Here you see a conscious cognitive strategy. So, okay, so us and other smart beasties like primates, here's another version of it, occurring in fish, in sunfish. And this was research done by that guy, David Sloan Wilson, that multi-level selection evolution guy who actually has done research in like so many different fields, incredibly creative guy. Here's a study that he did. Sunfish males, surprisingly, are quite paternal in there taking care of their eventual offspring. They're almost as good as Nemo's dad. And what you've got there is in this version, I'm not going to draw fish mating. Forget that. Okay, so the guy mates with a female, and she eventually gives birth to kids, and he helps take care of them. Now, instead, he mates with a female, and you, the savagely heartless researcher, puts him in the next tank over where he can see what's happening, and you fiendishly, at that point, drop down a clear plastic box right next to the female with another male in there. In reality, he's not mating with a female, but he's right there. And this guy who's stuck on the other side of the barrier, going out of his mind with jealousy and petulance and all of that immature stuff. And what happens is, after she eventually gives birth, this apparently being the female in this diagram, after she gives birth, he doesn't take care of the kids as much. There is no difference in any of the sensory cues because he, in fact, is the only one who mated. This one is kept inside. There's some sort of cognitive stuff even going on in a fish. Remarkable. So it's not just us. So we've got this broad realm, innate strategies. One's on very all or none sensory imprinting. And then there's the folks who think through it. We're in the mainstream of that, but we're not the only ones. So how do our brains do that? There's a part of the human cortex called the fusiform cortex, and what it's good at is recognizing faces, which is a remarkable thing. It specializes in recognizing faces, facial expression, degree of relatedness. You show someone a good portrait of someone else, and you will get that part of the brain to activate as if it was their face. You show a good cartoon of somebody, it will do the same thing, maybe with a little bit less confidence. This is the specialized part of the cortex that does facial recognition. Remarkable. You look at people with autism, and this part of the cortex doesn't do a whole lot. You show a non-autistic individual a picture of a well-known loved one. Fusiform cortex activates a whole lot. You show them a picture of someone they don't know a whole lot. It activates somewhat to a lesser degree. You show them the picture of an armchair, doesn't do anything at all. You take someone who is autistic and you show them, and you get the same low degree activation for all three. You know, mother equals stranger equals armchair. This in some ways is the core of what autism is about. You see that going on as played out in this part of the cortex. So that's really interesting. And then it turns out we're not the only species that has this specialized fusiform cortex. Primates have this as well, non-human primates. Sheep have it. It turns out pigeons, who could recognize pictures of each other, and why they would want to do that, God knows. But pigeons have a very proto version of a fusiform cortex. This seems to be part of what goes into this conscious cognitive strategizing of this is a face I've seen a whole lot more over the years, so this activates a lot more. This seems to suggest some sort of cortical specialization for recognizing individuals. More features of how humans do it. Something that humans are very good at, if they are human mothers, is recognizing the smell of their baby right after birth, and this has been shown to have a major histocompatibility component to it. In other words, that's not a purely cognitive strategy. There's some innate, instinctual olfactory signaling going on there. So a first bit of evidence that we are not just purely rational beasts at figuring out who we're related to. Babies, very shortly after birth, are already spectacularly good at distinguishing the smell of mom versus somebody else. How do you tell that with a baby? You take a newborn baby and you give them, like on this side, some armpit smell of mom, and there's some armpit smell of, you know, I don't know, Margaret Thatcher. And what you see then is that the baby, the newborn, is more likely, will spend more time turning its head towards mom's smell. That's how you know it. Newborn babies cannot distinguish between the smell of dad and any other male. It's not instinctual in that case. Newborn humans are doing some version of proximity to mom, vaginal fluid smell, whatever. This should be bringing up a question of, is there a difference in bonding in those early stages between vaginal birth, offspring, and cesarean? I don't know, but that suggests that should be happening, and maybe I should even find that out. Okay, Other features of it. Newborn kids, as we already know from a couple of lectures ago, can recognize the voice of mom. How? From all that resonance there inside the amniotic fluid as mom reads them War and Peace. And as we also heard, they can't recognize the voice of dad, not instinctual, in this case getting information on the sensory route, the amniotic environment as a good resonator for mom's voice. So we're now already getting a mixture here of some instinctual olfactory stuff, major histocompatibility complex stuff, some acquired, hardwired, sensory-driven stuff. Ooh, does this sound like the person whose voice I heard for those last 9 months? And then some cognitive stuff, trying to figure out who this relative is. Ooh, who did they come with to the party with? Who, all of that, the cognitive stuff. So already a suggestion that we're not just such pure cognitive machines. So where is that most interesting? And this is in a totally fascinating series of studies that were done over the years showing just how little conscious cognition might play out in us humans in some really critical realms, which is who you decide you are interested in mating with. Okay, so how does that work in humans? All sorts of different ways, but here's one way in which it does something interesting. And this was a classic study done by an anthropologist named Joseph Scheffer, and this is what he did. He studied people who grew up in Israel on kibbutzes. Kibbutzes, these are these traditional socialist sort of communes where none of them are like this anymore, but one of the early principles was one parent as good as another parent. All the kids are raised in these big communal bathtubs together, and it's one big sort of socialist Torah dance of everybody bathing naked together. And what Scheffer found was this very interesting thing, which is you are brought up in your age group. All the kids born this year are raised in the same communal group, and they take their baths together and they play together, and they've got the same, one parent takes all of them for one afternoon every Tuesday afternoon, and the next one comes for the next shift, and this big communal business there. And what Scheffer discovered was if you were raised in the same age group as somebody anywhere up to 6 years of age, you never ever wind up marrying them. And this was not with a sample, an appropriate sampling. This was a study he did of every individual who ever grew up in the kibbutz system in Israel. We appear to have a rule as follows. If this is somebody who you spent a whole lot of time with before 6 years of age, this is someone who you sure don't want to grow up and marry. Yuck, I mean, you love them, they're incredible, but they feel like my sister, they feel like my brother. Whoa, that would be totally grotesque. There's never been a case of people brought up during grow up in your first 6 years of life taking a whole lot of baths with someone, and you are not going to discover an amorous passion for them when they're 20 years old. They're going to feel like a sibling for you for the rest of your life. What is that telling us? We have a very non-cognitive strategy in there. Yes, who are relatives, who are appropriate people to mate with? Well, if we know that this is the daughter of mom's sister, then this person is not appropriate, and this whole cognitive, spend a whole lot of time naked with somebody, taking baths and playing patty cake and counting their toes the first 6 years of life, they feel like a sibling. Something very similar was then shown by Arthur Wolf here in the anthropology department, a different cultural version, traditional Taiwanese marriages, where there's some equivalent of that, and where either you wind up with your future spouse at some insanely early age and basically get brought up with them from infancy, or it happens later, and if you're brought up from infancy with them, you have a disastrous marriage later on because they feel like a sibling for the rest of your life. So what this shows us here is, yes, we are these wonderful, rational, cognitive machines. We've got all sorts of innate strategies and sensory imprinting stuff going on instead in us. We are not a whole lot fancier than hamsters. What does this set us up for? A topic that's going to be real important when we come to the lectures on aggression and cooperation and competition and all of that, which is if we spend a lot of our time figuring out who we are related to cognitively, or even more importantly, if we are malleable in the way these kids growing up in Taiwan or these kibbutzes, if we are malleable in who we feel related to, It is possible to manipulate us in lots of ways to feel more related to individuals than we actually are, or to feel less related to individuals. And the terms for these are pseudo-kinship and pseudo-speciation. And what we will eventually see is when you make sense of human violence and human cooperation and human aggression and all of that, we are so easily manipulated as to who counts as an us and who counts as a them. And one of the brilliant things militaries all the world over do, whether you're talking about clans with warrior classes all the way up to sort of state militaries, what you see in all those cases is a brilliant understanding of how to make non-relatives feel like they're a band of brothers and how to make them seem so different they hardly even count as humans. So this ability for us to be manipulated in these subliminal ways as to who counts as a relative will come back to haunt us big time in making sense of a lot of human social behavior. In other words, we are not purely rational. Okay, so what we will do on Monday is switch another bucket now to this field of ethology, which once again is trying to make sense of what behaviors are hardwired, what does environment do. You will see a completely different approach.
For more, please visit us at stanford.edu.